processing, as we call it, how this sort of cutting up of the RNA takes place, is something that Emmanuelle Charpentier demonstrated. She found a new small RNA molecule in the bacteria called tracer RNA. And she showed that this tracer RNA would bind to the long form of CRISPR RNA, and then together with two bacterial proteins, Cas9 and RNase-3, would cut the long RNA molecules up in small pieces. So now there was a piece of RNA corresponding to a previous infection. But how could this RNA piece actually protect against viruses? To answer that question, Emmanuelle Charpentier and Jennifer Doudna teamed up. And together the two scientists made a crucial discovery. They found that the tracer RNA that Charpentier found a year earlier, the CRISPR RNA, the small piece of CRISPR RNA, together with a Cas9 protein, formed genetic molecular scissors. And these scissors would use the CRISPR part of the complex and then search for virus DNA within the bacterial cell. And if there was a match where the RNA would match the DNA of the virus, then it would cut the viral DNA and disarm the virus. So this was a great discovery in itself, but the two scientists did not stop there. They wondered, could they make this system even simpler? Because now they had two RNA molecules and one protein. So what they tried to do was to fuse the two RNA molecules into one single RNA molecule, which they call a single guide RNA. And then they investigated if this single RNA, together with the Cas9 protein, could actually target and cleave virus DNA in the test tube, and it could. It worked very nicely. So they had now created a simple two-component system. But what they also did was that they started to make artificial guide RNAs. They changed the sequence of the CRISPR part of the guide RNA. And it turned out they could actually cleave almost any sequence of DNA that they would like to target. They had created a programmable machinery, programmable genetic scissors that could be used to cleave DNA in the test tube.